Dear ladies and gentlemen, my name is Philip Rosenstock. I'm product manager for infectious serology at EUROIMMUN Germany, and today I'm going to talk about SARS-CoV-2 vaccination control analysis, and I'm proud to present you our latest developments in the for SARS-CoV-2 antibodies and T-cell responses. Before we start talking about the analysis itself, I'd like to tell some or mention some points about the background. So why is vaccination control now frequently requested? So first of all, you might have heard or it came to your ears that the vaccines itself do not show a complete vaccination efficiency. That means protection rates between 70 and 95% were described for vaccines like Moderna, AstraZeneca and BioNTech Pfizer. Thus, there is a personal interest to check if an individual has reached a protective antibody titer. Second, there is of course, due to this ongoing discussion, big uncertainty if the vaccination has worked, and thus this vaccination control might help to reduce these uncertainties. The next point is that elderly patients or people without a high immunocompetence are vaccinated with high priority. And these people are, of course, especially at risk for an infection. However, the immune system might not be working properly, which is why, and this is what I'm going to show also later, especially in this group, there's not a 100% efficiency, and this makes an immune status control of high interest, or it underlines the need for immune status control. And last but not least, we have the nursing personnel that gets in touch with patients that are of risk to develop an infection. And of course, in this nursing personnel, we need to be sure that the vaccine worked efficiently. So that underlines that we of course need to have an immune status control after vaccination. But the central question now is how to control the immune system after vaccination. But before we talk about the immune status control, I'd like to also show some backgrounds of the immune system that is triggered after the vaccination. So the vaccine itself, or parts of the vaccine itself, or the mRNA are leading to the presence of spike protein. And this spike protein is then taken up by antigen-presenting cells, and these antigen-presenting cells are then presenting subparts of the spike protein to immune cells. So, of course, in the focus is always antibodies. The secretion of antibodies is, of course, the most prominent immune marker. Antibodies are secreted by B cells, but B cells cannot be switched on alone. So they need also T helper cells. And this interaction highlights that the immune system has two branches that are stimulated after vaccination. And of course, these two branches go hand in hand. So antibodies are, of course, mediating neutralization. They are supporting the pathogen uptake by immune cells by marking the pathogen itself, and they are able to activate the complement, and the complement then triggers further immune reactions. However, there's also a branch of the immune system, a cellular branch based on T cells that acts alone, which is the initiation of cytotoxic T cells. And the function of these cytotoxic T cells is that they can kill infected cells specifically. So cells that are infected with SARS-CoV-2, so that the infection cannot spread any further. So the virus reproduction is interrupted. But also here, the T helper cells are needed to efficiently stimulate the cytotoxic T cells. So it is a close interaction of cellular immune system components that lead to the secretion of antibodies on the one hand and of cytotoxic T cells on the other. And if we now want to detect the immune system in a holistic approach, of course it's not sufficient to detect antibodies alone or to detect T cells alone. It's rather interesting to check both branches of the immune system, at least in patients with unclear immune reaction. So this was just a background to highlight the necessity of the immune system control, and I also showed that antibodies and T cells are to be monitored. But what's the appropriate target to be detected? So what kind of virus component do we need to check, to test for? So the currently registered vaccines that are available in the European Union are BioNTech, Pfizer, Moderna, AstraZeneca and Johnson & Johnson. And what they have all in common is that they are encoding or carrying the spike protein that contains the receptor binding domain that interacts with the human host cell. So only few exceptions are based on full virus preparation. One of the most prominent is the Chinese Sinovac. That means for the detection of immune response to the currently registered vaccines, the detection of the anti-spike immune response is required. So now I'm happy to show you or to present to you the trio of vaccination control analysis EUROIMMUN can provide. So first of all, most prominent, that's the regular quantitative IgG antibody assay that contains the spike subunit S1. So this assay, the SARS-CoV-2 Quant-T-Cell IgG ELISA, contains the appropriate antigen to detect immune responses after vaccination. It has a complete, fully quantitative evaluation. It correlates exactly, excellently to the WHO reference preparation, and it can be used also with dried blood spots and not only with fresh plasma or serum, which makes the assay especially interesting for remote analysis. One of our latest developments is the SARS-CoV-2 NeutraLISA, and as the name indicates, it's a surrogate neutralization assay. And the neutralization assay is currently considered as the gold standard for the detection of antibodies because it shows the efficiency of antibody reaction. So this NeutraLISA is based on the RBD-ACE2 interaction and therefore can directly be linked to the occurrence of neutralizing antibodies. So it gives information about the functionality of the antibodies, and in contrast to the regular normal neutralization assays, there is no increased security requirement necessary, and high numbers of samples can be analyzed. And last but not least, I'm going to present you our new Quant-T-Cell SARS-CoV-2 assay. This assay is intended to be used for the control of the cellular immune system. So it is a measure to detect the stimulation of T cells after both vaccination and wild-type infection. But today, vaccination is of course in focus of my talk. And this assay can be used as a supplement to antibody detection methods. So, as mentioned before, I'm going to start with vaccination control analysis with a regular immunoassay, which is now the SARS-CoV-2 Quant-T-Cell ELISA. So, as mentioned before, we need to offer the spike protein or subcomponents of the spike protein in order to detect antibodies after vaccination. And this assay, the new Quant-T-Cell ELISA, is based on the well-known S1-based EUROIMMUN ELISA. So it uses the same microtiter plate, the same cutoff, but the assay is extended by a huge set of calibrators so that a fully quantitative result can be obtained, which is of course interesting in case dynamics or comparisons or statistics are going to be established. And it can be used for serum, plasma, and DBS testing, so it can also be used for remote sampling. As mentioned before, we have been able to correlate this assay to the WHO international standard preparation, so that a standardization and the possibility to compare the results to other immunoassays is possible. And it is quite easy because we have a great linear correlation between our relative units that are given by the assay and the now established binding antibody units, BAU per ml. The conversion can be performed by just multiplying the results by the factor of 3.2. Why can't we use the term international units? Why do we need to assign the results to these binding antibody units, BAU per ml? So this is defined by the WHO and the NIBSC. They stated that international units are exclusively to be used for neutralization tests, so for the detection of neutralizing antibodies, while binding antibodies are to be referred to these binding antibody units, BAU per ml. However, the numerical value is identical, so that there is a direct correlation between international and binding antibody units. So now, how does the assay perform in vaccinated individuals? So there are first publications available. This is a picture taken from Müller et al. And they checked the age-dependent immune response after the vaccination with the BioNTech Pfizer vaccine with our SARS-CoV-2 Quant-T-Cell ELISA. And what you can see is that after the second dose was given, we have extraordinarily high antibody activities that are detected. Of course, this is good news when talking about the efficiency of the vaccine. It seems to be very highly efficient. However, what you also see here is that the vaccine success is different in two different age groups. The blue ones are younger people, while the green colored groups are older people. In picture A, you see the immune response to the antibody secretion after the first dose. In picture B, it's after the second dose. And what you can see is that the mean value in elderly people is much lower. And also the seroconversion is reduced in elderly people, as you can see in picture C. So there are some patients or some persons that do not develop positive antibody results, indicating that the vaccination was not sufficient in these patient groups. So that means, of course, we have a difference in the mean antibody value and we have a difference in the individual antibody values. So when can we assume the vaccination success? And unfortunately, there is no vaccination cutoff available so far. So these vaccination cutoffs need to be established on epidemiological data. So that's long-term experiences, and we do not have long-term experiences because this disease is new and the vaccination is also pretty new. Or it is based on huge trial studies that are initiated by the vaccine manufacturers, but also these data are currently lacking. So these thresholds or cutoffs for immunity are usually released by independent institutions or the manufacturers itself. However, there is no correlation, so there is no immune threshold that was released so far. So how can we currently assume a vaccination success? So this is only possible on the basis of correlation to a neutralization assay. And there are also already some data for our SARS-CoV-2 Quant-T-Cell ELISA that show a good correlation to neutralization assays and that show that the cutoff that was validated for the Quant-T-Cell ELISA is correlating excellently to cutoffs from neutralization tests, so that the results can be correlated easily. So on the upper side of this page, you can see two internal correlation studies, one against a commercial surrogate neutralization assay and one against a real plaque reduction neutralization test. And you can see that the correlation is excellent. On the bottom of the page, you can see the borderline range, so defining the cutoff, and the correlation to neutralization assay that was released by Rubio, Azzero et al. And you see that the cutoff that we have defined is pretty much identical to the correlation between positive Quant-T-Cell and the neutralization titer that was found by Rubio, Azzero et al. And also the correlation to a surrogate neutralization assay shows that the cutoff was selected quite well, so that if you receive a positive result with a Quant-T-Cell ELISA, you can be pretty sure that the antibodies are neutralizing. And we are quite often asked if there is a discrimination possible between antibody activities that are triggered by wild-type infection and those that are triggered by vaccination. And there is currently no sole marker that can be used to distinguish these antibodies. However, please remember that the majority of or all vaccines that are currently registered in the European Union are based on a spike protein. In contrast, after wild-type infection, of course, also antibodies against the nucleocapsid protein are triggered. And if you compare both results, so spike IgG and nucleocapsid IgG, from the resulting pattern, you can discriminate if the antibodies are triggered by vaccination or by wild-type infection. Because after vaccination, we do not accept any nucleocapsid protein titers, while after wild-type infection, both markers should be positive. This was described by Lichtenfeld et al. And also by Dershuk et al. And they used our S1-based assay and our nucleocapsid-based assay in order to create the figures that are shown here. So that means, in summary, if you combine our assays, the spike S1-based SARS-CoV-2 ELISA or SARS-CoV-2 Quant-T-Cell ELISA and our SARS-CoV-2 NCP ELISA, you can mediate or you can differentiate between antibodies that are triggered by vaccination or by wild-type infection. Last but not least, I'd like to highlight the use of DBS, so dried blood spots. These dried blood spots can easily be obtained by the patient itself. It's just puncture of the fingertip. The blood is then put onto a filter card that is provided by EUROIMMUN, and you can easily ship these samples then to a lab just via the normal postal service. So the assays and our automates were validated for the use of dried blood spots, and we also offer a set box that contains all necessary materials that are needed to either take the sample by the patient himself or herself or by a lab technician and so on. So this assay or these samples make it easy to screen a huge number of patients. So the next topic I'd like to talk about is our new NeutraLISA. So as mentioned before, that's a surrogate neutralization assay in an ELISA format. So neutralizing antibodies are directed against the receptor binding domain that is present on the spike S1 subdomain. This receptor binding domain interacts with the AC2 receptor on the host cell. In case you have antibodies against the receptor binding domain, it covers the receptor binding domain, so it cannot bind to the ACE2 on the host cell anymore, so that infection is stopped or not possible at all. And these antibodies are considered as the most promising marker of immunity. So if you ask yourself, so why should I use a neutralization test, or why should I use an ELISA like our Quantivac ELISA? Aren't these two assays giving the same results? This is true only in parts, because there are some different characteristics behind. So the neutralization assay detects just functional antibodies, only those that prevent RBD-ACE2 interaction. However, there might also be antibodies against the spike protein that are not mediating the protection, but that are detected in a normal immunoassay. Maybe it's just antibodies that, or these antibodies are directed against a rural epitope of the spike protein. And in a neutralization assay, all kinds of antibodies that mediate neutralization are detected. So it does not carry a specific, let's say, for example, IgG conjugate. Since IgA antibodies have a high neutralization capacity, also, of course, these antibodies are important to be detected. And what we have learned from some unpublished studies is that the IgA response is very high after vaccination with, for example, the BioNTech Pfizer vaccine. And last but not least, it's not only antibody binding, but it's also the strength of the antibody binding that plays a role in neutralization. Because antibodies of low affinity or avidity might be displaced by ACE2 because it's binding more efficiently. And this, since in a normal ELISA format we don't have this competition, this might not be detected. In contrast, the ELISA detects all antibodies against the spike protein, not only those that are mediating neutralization. However, it's possible to distinguish between IgA, IgG, and IgM so that you can monitor individual dynamics. And also here you are able to detect antibodies of lower affinity because we don't have any competition between ACE2 and the antibodies that bind to the spike protein. So the NeutraLISA, the SARS-CoV-2 NeutraLISA that is to be released by the end of March 2021, acts like a neutralization test, but in a microtiter plate. It's not the first assay that's on the market, and there are some few others, but we have some advantages here. So what the surrogate ELISA all have in common is that they are giving similar results like a neutralization assay. However, you do not need to handle a virus stock. So the virus stock would be, there's a need to be handled under biosafety level 3 conditions. So this is not necessary for this assay. So it's a standard laboratory procedure. It's like in ELISA, and it can be, of course, processed automatically with all EUROIMMUN devices, or also maybe other devices like Dynex, DSX, or similar. The results are available within few hours, which is of course different to a neutralization test that needs this incubation for several days, and that of course makes the ELISA much more cheap compared to a real neutralization assay. So what's the background? So what's the interactions that we are going to detect? So in contrast to several other surrogate neutralization assays, we use coated S1 as on the solid phase. Then we add a mixture of the patient samples that was pre-incubated with biotinylated ACE2, so the host cell receptors. If there are neutralizing antibodies directed against the RBD present, we can prevent the ACE2 binding. In case there are no antibodies against the RBD, ACE2 can still bind. This ACE2 is biotinylated, and in a secondary reaction, you can detect this biotinylation via our substrate, which is a streptavidin labeled with HRP. That means in case you have neutralizing antibody, the signal is reduced, while in case you have no neutralizing antibodies, you have a full signal indicating the ACE2 binding. So the procedure is just like a normal ELISA. The only thing that is different is that you need to pre-incubate your sample with a special dilution buffer that contains the soluble ACE2. However, this is also, if you use our EUROIMMUN devices, automatable. So then we have some specific incubation periods, like used for our normal ELISA, with conjugate incubation, substrate incubation, some wash steps in between, and then you just receive a single result. Single result, yeah, well, that's a little bit to be explained. What you need to know is a reference value. You need to know how huge the inhibition of the ACE2 as one interaction is for the individual patient. That means you need a reference value, and this reference value is a blank. So that means you have one control without any antibody that just contains ACE2, and this shows you the full amplitude, so the interaction between S1, RBD, and ACE2 without any interference. And each individual patient is then referred to this result. And the result is a % inhibition. So what is done? You take the OD of your patient sample, divide it by the blank, so the reference value, subtract it from 1, and what you get out is a % inhibition. For example, 70% inhibition or 30% inhibition. Positive control is included, negative control is included, and a cutoff control will also be included. So we have been able to already check or correlate the ELISA with our previously described Quantivac ELISA, and as you have seen in the previous slides, we have a very, very good correlation of both assays, of both technologies. But also with a real neutralization assay, we were able to show 100% agreement between both techniques. So this is how the assay is set. It's just, it looks like a normal, regular EUROIMMUN ELISA, so it's a microtiter-based assay. However, the only thing that is different is the sample buffer, because it contains the soluble ACE2. And the rest is more or less identical. And of course, as mentioned several times before, you can perform the ELISA either manually or automated with our analyzer or EUROLab workstation ELISA devices. So now, last but not least, I'm going to talk about the T-cell assay. So this is our SARS-CoV-2 Quant-T-Cell assay. As mentioned before, well, the detection of antibodies is kind of a gold standard. However, it might not be sufficient to only detect antibodies. Why is that? That's going to be presented in the next few slides. So again, I'd like to repeat some basic information about the immune system. So the immune system acts in a concerted action to clear a pathogen after infection. So the pathogen itself is targeted by antibodies, and infected cells are killed by cytotoxic T cells. And we have the T helper cells that help the stimulation of B cells and that help to stimulate these cytotoxic T cells. And in order to efficiently stimulate the immune system, both antibodies and T cells need to be stimulated. So immunity requires two branches. Why should we detect the cellular immune system additionally to antibody responses? So there are two reasons. First of all, diagnostic reasons. So detection of previous infection. And second one, which is the focus for today, determination of immunity also after vaccination. But first, I'd like to have like to lose some few words about the diagnostic reasons. So why should we detect the immune system on the basis of the cellular immunity, but not only on antibody testings? So the reason is that some patients, so it was described, so up to 10%, do not develop IgG antibodies after an infection. That means these people remain seronegative for antibodies, and of course here the cellular immune system might be an additional marker. Second is that antibodies get lost after a certain period. So it was described that the half-life of anti-nucleocapsid IgG antibodies is, for example, much shorter than those of the anti-spike IgG response. However, also here for the spike IgG, the period of positivity is not, let's say, not too long. It's not that it is to infinity in any cases. So here it might be necessary to detect, or might be helpful to detect, the cellular immune response, since memory cells will be present over a longer period. And this was already published in several publications. Now coming to immunity, also after vaccination. Still, I've to mention that the correlate of immunity still remains unclear. However, it is assumed, and it is also described, that the cellular T-cell immunity plays a major role in the mediation of immunity. And many vaccines, most prominently the AstraZeneca or the BioNTech vaccine, trigger both the humoral and the T-cell immune response. And also publications, for example, by Shahin et al. That's the head of the company BioNTech, were able to show that vaccines trigger T-cell responses efficiently. So if you want to detect immune success and you aren't able to detect antibodies, the additional testing of T-cells might be of help. And how to detect the activation of T-cells? Like antibodies, T-cells are specific for a defined epitope. For example, SARS-CoV-2 spike epitope. And it's before stimulation, naive T-cells are circulating in the blood until they are going to be activated in case they met the specific protein. Then they are going back to the lymph node, and there they are going to be stimulated efficiently, also with the help of T helper cells. And then the activated T cells are circulating in the body and mediate the immune response to SARS-CoV-2. And this interaction or this activation can be detected by interferon gamma release, which is an activation marker. And this is also to be determined here in our Quant-T-Cell test. So what kind of material is needed to check the cellular immune response with the Quant-T-Cell ELISA? So it's whole blood, so you need fresh lithium heparin tubes with whole blood that has to be stimulated within 16 hours after sampling, because the cells need to be fresh. If you store it too long, they are going to be degraded and cannot be activated efficiently anymore. The stimulation requires 3 tubes. Each tube is filled with 500 microliter, incubated over a longer period, and then the plasma is prepared, and this plasma is then to be analyzed for interferon gamma release in an ELISA. After the plasma is separated, the sample can be stored for a longer period, of course. So why do we need 3 tubes for one patient? So first is the stimulation tube. This one is needed to show that the sample still can be stimulated also after, let's say, a storage of 16 hours. So it contains an unspecific stimulant. Next one is a blank tube. We always have a kind of a background reactivity of T cells that is differing between patient and patient. So it might be lower, it might be higher. However, to have a reference value, we need to detect the blank for every patient. And then we have a tube, and this tube contains specific stimulation reagents. It's peptides of the spike protein. And this is then the specific signal that is going to be analyzed. However, it needs to be referred to the blank tube. So how does it work? Again, you take fresh heparin tubes, you transfer 500 microliter each to the 3 tubes, invert it, incubate it at 37 degrees for 20 to 24 hours. Then you use centrifugation or let's stand in order to have serum. And this serum can be then used immediately for interferon gamma analysis, or it can be stored at minus 20 or 4 degrees. So the interferon gamma, this is what's going to happen in the tubes. So in the specific tube that is coated with S1 spike peptides, the T cells that are specific for SARS-CoV-2 are going to be stimulated. The secreted interferon gamma is then analyzed in an ELISA. So this is just like a normal interferon gamma ELISA that contains an antibody directed against interferon gamma. And this interferon gamma is then detected with a secondary antibody that is linked to an enzyme that mediates a color change. So this is how the assay looks like. It consists of 2 different products. One is the stimulation tube, and the other product is an interferon gamma release assay. So the data evaluation is as follows. So a positive result indicates an effective vaccination. As indicated before, there is no immunity threshold also here. I indicated that for the antibodies, and this is also the case for the T-cell response. However, there are some independent publications that are going to be released soon, and they show a very good performance also after vaccination. So, and they will state that this assay is an appropriate tool to detect vaccination efficiency. And however, the official immunity thresholds are not available. So then, with this last slide, I'd like to finish my talk. Thank you very much for your attention, and I would be very happy if I was able to gain some interest in our products. Please let us know if you have any questions on this essay. We are happy to share further information with you. Thank you, and bye-bye.